go through the information here. 12.7 Structures of DNA. Okay, so first of all, you need to know DNA is in our cell nucleus. Okay, so nucleus. So the genetic information is inside. So what is DNA? So DNA are polynucleotides. Polynucleotides. So of course this is a polymer. Okay, this is a polymer, and then the monomer is nucleotide. The polymer is polynucleotides. Okay, so we will look at the structures afterwards. So let's see what else. So they said here, make by condensations, polymerizations of nucleotides. So from here we have to understand that when they say make by condensation polymerization, and that means a small molecule will be taken away when they form the polymer. So they may remove the H2O or the HCl, for examples, when they form polymer. And polymerizations of nucleotides. So from here we also know that the monomer is nucleotide. Monomer means single unit. So when they join up, we call it polymer. So the polymer is polynucleotide. Okay, now let's move on. Functions of deoxyribonucleic acid, which is DNA. So what's the function? Of course, it is related to the genetic stuff. Okay, so now let's see what they put here. Can make copies of itself so that genetic information can be passed on from generation to generation. So this is the DNA, the functions of DNA. So you can just copy the information on the DNA and then just pass on. Okay, so for example, during cell divisions, so the cell is separated into two units, and at the same time the DNA is separated also. I mean the DNA is copied and then is formed into two new sets. Okay, so that is basically the functions, what happens during cell divisions. Okay, now let's see the next point. Contains sequence of bases that form genetic code used to synthesize proteins. So in DNA, there are bases, sequence of bases. Okay, so you can see base, okay, sequence of bases. So examples of base is like A, G, T, C. Of course, you can see here we have double ring bases A and G. We have single ring bases T and C. So these are the bases. So the bases form the genetic code. They arrange in different way and form genetic code, and that genetic code is used to make proteins. So for example, let's say to make hair, so either it is straight hair or curly hair, so the DNA contains genetic code that will make the hair either curly or straight hair. So it is in the genetic code. And then next point, nucleotides in DNA are made up of, okay, nucleotide, remember nucleotide is the monomer. So the monomer itself has three components, as you can see the diagram at the side here. So the first component is the round shape, then the pentagon shape, and then the rectangle. So these are the three components that make the nucleotides, and nucleotides is the monomer used to form the polynucleotides, which is DNA. Okay, so now let's see what are all these, the components here. So the first one is called a sugar. The sugar is called Deoxyribose. Deoxyribose. This is a pentagon shaped. Okay, it's a pentagon shape. So this is the pentagon. Okay, pentagon. And then the second component is the phosphate group. Phosphate group is represented by the red circle over here. So this is the phosphate group. Phosphate group is the red circle. Okay, so phosphate, of course, you should expect phosphorus and oxygen there. Phosphate. Okay, and then the next one is nitrogen containing base, which is the rectangle over here. So just now I have gone through nitrogen containing base, where I said there are double rings, which is adenine, guanine. Single ring we have thymine and cytosine. So these are the bases, and remember that the function of the bases here is to form genetic code, so that they can make different kind of proteins. Okay, now let's move on. Complementary base pairs. Okay, so that means if the base is going to they are going to pair up actually. So they are going to pair up. So the structures is like this. Let's take a quick look first. Our DNA, right, is made of two polymer. It's made of two polymers, which is staying side by side. It is something like this. As you can see, this is one polymer. Okay, this is one polymer. Okay, so where let's say I put a circle like this. This is one polymer. And this is another polymer. So there are two polymers. And these two polymers, right, they are linked together. Okay, they are linked together by using the base. You see the base is the rectangle that we discussed through just now. The base is the rectangle. And then the base you pair up and they form certain bond here, so which is called the hydrogen bonds. So they don't simply pair up, So if you have base adenine, so you have to pair up with thymine, A and T, yeah. So it's one pair, and if you have guanine here, then you have to pair up with cytosine. And if you have thymine, then you have to pair up with adenine, and adenine, thymine, cytosine, guanine, cytosine. Okay, so they have to pair up in that particular way, where A must pair up with T, or the other way around, T with A. G must pair up with C, or the other way around, C with G. So they have to pair up in that way. Okay, so how I remember it is, I look at the letter, So basically A and T, right, you see the letters are all formed by straight line, So straight line and straight line letters are one pair. And if you compare with G and C, right, there is a when you write G, you have to draw a curve, So they are round shape after all. So round shape. G and C are round shape. This is the second pair. I remember it in that way. And the other thing is, if you can see, let's say I zoom in a little bit. Okay, so I zoom in a bit. As you can see, another thing, for adenine and thymine, there are two hydrogen bonds, 1 and 2. Guanine and cytosine, they have three hydrogen bonds. To link them together. And how I remember it is, if their letter is made of straight line, as in A and T, so we just need two hydrogen bonds to link them. And if there are G and C, which is made of round shapes, so I think of example like ball, ball can easily roll away, so we need 3 hydrogen bond to fix it in that position. I just imagine in that way, like, they can easily roll away. So need more hydrogen bond to fix them in the position. So it helped me to remember it. So remember the facts here. A and T is one pair, G and C is one pair, A and T has 2 hydrogen bonds, G and C has 3 hydrogen bonds. So you have to remember it. Okay, now let's come back to this side. Okay, so this is what we discussed just now. A and T is the first pair. Okay, of course it can be the other way around. T with A also can. So just now we also discussed it. A and T form 2 hydrogen bonds. Okay, so you can see it here. 1 hydrogen bond, 2 hydrogen bond. Okay, 2 hydrogen bonds. So the first one is between H and O. The second one is between N and H. 2 hydrogen bonds. G and C, they form 3 hydrogen bonds. So the first one is here, 1. The second one is here, H and N. The third one is H and O. Okay, so 3 hydrogen bonds for this and 2 hydrogen bonds for this. Okay, so now let's move on. The other side. Okay, so remember one thing before we move on to the next slide, because this chapter is polymerization chapter, so we need to know that these whole structures is called nucleotides, and nucleotide is the monomer. It's nucleotides. Okay, now let's see the next part on the right hand side. Structures of DNA. Structures of DNA. Okay, so just now we already go through the pictures. So you see there are 2 polymers here, and we call it double helix. Double helix. So in actual situation, they are not just straight polymer, they are twisting around like this, they twist around like this, like something like a spring, and we don't call it spring, of course we call it double helix. So that's how you see the terms here. Double helix means the 2 polymers that twist around, and at the meantime you can see also the base are paired up accordingly, you see. This one with this one, this with this, this with this. So they pair out accordingly, like what we discussed just now, A and T and G with C. Okay, now let's see the words here. Structures of DNA consist of a double helix made up of 2 strains. 2 strains mean 2 polymers kept in place by hydrogen bond between the nitrogen bases. So remember, the base that we discussed just now is also called nitrogen bases, as you can see here. Nitrogen containing bases. So you see the base here has nitrogen, you see here and here nitrogen. So we call it nitrogen containing bases or nitrogen bases. So let's move on. Yeah, so they found hydrogen bonds. The base found hydrogen bonds. Now, next point, as you can see here, each strain has a backbone. So the strain here is a polymer, remember that. Each strain has a backbone of alternating sugar and phosphate units. This is on the outside of the structures. Okay, so it means so you look at the picture again. Okay, so the backbone I put in blue color. This is the backbone. Okay, this part is the backbone. This part is also the backbone. So what we can see from the backbone is, this is a backbone. This is another backbone. So you can see they alternate. So as in here, you see phosphate followed by sugar, phosphate and sugar, phosphate, sugar. Okay, and then phosphate, sugar, phosphate, sugar, phosphate, sugar. But this side, right, it is the other way around. It's like upside down already, you know. We read from the bottom: phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and phosphate, sugar. So you can see they are the other way around. Huh? This one is like here to the bottom, but this one is like the bottom to the top. It's something like that, you know, the other way around. Okay, so in terms of 3D, it look like this. The blue color part, which is backbone, is in 3D is this strain. This gray color strain is the backbone. Okay, so this is one strain, and you can see how they name it. This is like 3 prime, 3 prime come all the way and here, so end up with 5 prime. So 3 prime to 5 prime. You can see something like that, 3 prime to 5 prime. So what is that? I will explain. Okay, so 3 prime to 5 prime for one strain. Okay, so the other one you can see here, 5 prime, turn, and end up here, 3 prime. So one strain is, or one polymer is 3 prime to 5 prime, another polymer is 5 prime to 3 prime. Okay, so let's see how. Okay, let's see how the 3 prime and 5 prime is formed, you know, how we see that. Okay, so let's put it here. Okay, so I draw a structures of the sugar, then it will be easier to understand. So the sugar is pentagon shaped, as you can see. As you can see here, the blue color pentagon, this one. Okay, pentagon is pentagon shaped. Okay, now we need more information on this so that we know what is 5 prime and 3 prime. So I draw it here. So let's take a note, extra note. So the pentagon shape is having oxygen, carbon, 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 and then carbon again. Okay, and so on. So this is the sugar, which is in pentagon shape. Okay, there is one more carbon here. Okay, one more carbon over there. And after that, okay, now I put numbering for you. This is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So we need to understand the 5 prime, 3 prime, right? So it's related to the carbon 5 and the carbon 3. So first of all, I put this information. Phosphate group is linked between the carbon atom 3 of 1 pentose sugar and carbon atom 5 of another pentose sugar. Molecule. Okay, so this information is enough for us to understand the situation already. Okay, so which means what? Which means let's see. Okay, I zoom in the picture over here. Okay, let's understand it. Okay, now just now I already explained where the top one is oxygen. This is oxygen. This is carbon 1. Okay, so I put the numbering. Carbon 1, carbon 2, this is carbon 3, this is carbon 4, carbon 5 is somewhere here. You see there is a twist over there. This is carbon 5. So this is oxygen. Okay, so according to the words that I wrote just now, I said a phosphate group is linked between the carbon atom 3. Okay, so that means phosphate. You see this phosphate? This phosphate is linked to carbon atom 3 of 1 pentose sugar and carbon atom 5 of the next pentose sugar. That means you can see the phosphate group linked to carbon 3 and then linked to carbon 5. Linked to carbon 5. Okay, so this is carbon 5 of another. Carbon 5 of another pentose sugar. So you can see something like that. So phosphate link in that way. So phosphate linked to carbon 3 and phosphate linked to carbon number 5. So that is the idea. And what is 5 prime and 3 prime? Okay, so if you see this backbone, right, having carbon 5 on top and no more connection anymore after carbon 5 and the phosphate, then this is called 5 prime. So this backbone is 5 prime. Backbone is 5 prime. Okay, then you go all the way down when you reach here. Okay, so again, carbon 1, 2, 3, 4, 5. This carbon 3 is supposed to have phosphates below of it, but there is no more phosphate that stick to it anymore. So this is called the 3 prime. This is the 3 prime. Okay, so from here you can see this strain or this polymer is 5 prime to 3 prime. And how about this? Okay, so when you look at this, right, you have to put it in the opposite way. So first you need to know, because they are upside down, so you have to know this is the oxygen. Okay, this is the oxygen. So carbon 1 is here. Carbon 1 always linked to the base one. As you can see, as in here also, carbon 1 linked to the base. So you can see it here, carbon 1 linked to the base. Okay, so if that is the case, so my carbon 1 should be this one, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and then phosphate. So 1, 2, 3, 4, 5, and then phosphate. Okay, now as what we discussed just now, these carbon 5, after linking to phosphate, then no more connection already. No more already. It's the end of the backbone already. So this is called the 5 prime. Okay, the 5 prime. This is called 5 prime. 5 prime, this backbone. This end is called 5 prime. And if we go upwards here, so likewise, the numbering will be the same. Carbon 1 always linked to the base. 1, 2, 3, 4, 5. Okay, so this is where the carbon 3 doesn't link to any nucleotides anymore. So that is the end of the string already. So this is called 3 prime. Okay, so this end is called 3 prime. So that is how we understand 5 prime to 3 prime and 3 prime to 5 prime. So they are the other way around. Okay, so we discussed this already. Now let's move on. Okay, so we move on. This point, the here, the 5th carbon of one nucleotide, 5th carbon, okay, which refer to the carbon here that link to the phosphate, link with third carbon of the next nucleotide. Okay, so which means as in here, the fifth carbon. Okay, example here, the fifth carbon link with phosphate, then link with the third carbon. So that's why the note says the fifth carbon link to the third carbon through phosphate, no? Through phosphate. Okay, so now let's look at the notes again. Okay, we look at the information here again. The fifth carbon of one nucleotide links with third carbon of the next nucleotide. Two ester links with hydroxides of sugar. Okay, so it's ester link. Ester link means COO, no? COO. So COO. Forming sugar phosphate backbone on the outside of the structures. So we call this outside of the structure. You look at 3D, you know, because inside is the pairing of the base. Pairing of the base is inside, so the backbone is outside. Okay, so then next point, nitrogens. Okay, this point, nitrogen's bases are positioned between two sugar phosphate chain at right angles to the backbone on the inside. So you see this is inside. Even in 3D structures, you can see this is inside. Okay, the basis is inside. Okay, so we continue. At right angle to the backbone on the inside, filling space between two strands so that the helix has a regular shape stacked above each other. Okay, so it's something like this, stacked above each other. It's like they are twisting around each other. Okay, so each strand has a direction. There will be a free fifth carbon in one end, so which is this one, the free fifth carbon, and a third carbon on the other end. Third carbon on the other end. So it's 5' to 3', something like that, or the other way around. So two strands run in opposite directions. We call it anti-parallel. Anti-parallel. So that means they are talking about this. So which is this one? One strand is 5' to 3'. The other strand is the other way around. It's 3' to 5'. So these structures is called anti-parallel. Okay, so we move on here. The structures of DNA is stabilized by hydrogen bonds between base pair, okay, which is here or here, 2 hydrogen bond, 3 hydrogen bond, and so on. When the wall forces between one base pair and the next. So even here to here, there is when the wall forces that stabilize them. Okay, so we have two intermolecular forces that stabilize the DNA, hydrogen bonds and van der Waals forces. Okay, so now let's move on. Okay, so this part, 12.8, DNA replication. DNA replication, that means how they copy the genetic information, but we just need to go through briefly only. So let's see. Process of copying DNA during cell division. So when one cell wants to break down to two cells, they need to copy the DNA information and make it into two sets and separate the two sets into two cells. Okay, so that is the process of copying DNA during cell division. So the process is called DNA replication. Okay, so semi-conservative replication. Semi-conservative replication. So what is that? So let's see the picture first. The picture will help us to understand. In a cell, the DNA is double helix. where in 3D they twist around, but in 2D it's just like this. Okay, so when the cell is going to divide into two, so the DNA will just break down into two strains. That means the polymer will separate, which means the hydrogen bonds is broken down in between. Okay, so after that, once they're broken down, so you see one of the strain, they put it up here, and another strain they put it down here. So the strain up here will form new. They are trying to attract the new base accordingly. So A will pair up with T, C will pair up with G, G will pair up with C, A will pair up with T, T will pair up with A, and then the bases that come to this part, and at the end they form a new polymer. This is one side. Okay, so another side is from the right hand side. The right hand side, something similar happens. If this is T, it will pair up with A, G pair up with C, C pair up with G, T pair up with A, and A pair up with T. So once they pair up already, then it will form a polymer. So that is how they form two sets of genetic. Okay, so we call it semi-conservative because you see this is the original one, right? So semi is like half of the set is the old set, and another half is the new one. So likewise for the other cell, half of the genetic set here is the old one, and another half is the new one. So that's why we call it semi-conservative. Half is the old one, half is the new one. Okay, it is not to say one cell take both of the old one, the other cell take both of the new one. It is not like that. So it is each cell will maintain half of the old one. So that is called semi-conservative. Okay, so let's move on. Okay, here. So after looking at the picture together with the explanation, it will be easier to understand the words here. So hydrogen bonds and van der Waals forces between base pair in parts of a DNA are broken, and which is here, the breakdown. Before that, they are like joined up together, and this part of the double helix unwinds. That means initially they are double helix. Double helix means they twist around, so that is winding up. So now they need to unwind. So that means they don't want to twist anymore. They want to separate and become a straight chain. Okay, so now here in the nucleus, there are nucleotides. Remember, nucleotide is the monomer. So in the nucleus of the cell, it has nucleotides, free nucleotide, haven't joined up yet, to which two extra phosphates have been added. So we call it nucleotide triphosphate. Okay, nucleotide triphosphate. So this is a structures in the nucleus. So the bases of the nucleotide triphosphate pair up with complementary bases on the old strain, and hydrogen bond and van der Waals forces form between original and new strains. So they are trying to say this thing, So once they open up, then this base will come here. This base is called the nucleotide triphosphate. So which will come and pair up accordingly. So once they pair up, then they will form hydrogen bonds, then they will form van der Waals forces in between. And there is enzyme that make this happen. So the enzyme catalyze the polymerization reactions. So each new strain contains a sequence of bases that is complementary to the original strain. Same with the original one, like here 80, you see this one, the new one is also 80, this one, the new one, 80 also, same one. These two sets is 100% same with the original one. Okay, so as in here, the example original strain is ATGC. Okay, so then new strain will pair up accordingly. A pair with T, okay, A pair with T, T pair with A, G pair with C, and so on. So they pair up accordingly. Okay, now let's move on. Significance of hydrogen bonding in replication of DNA. So very important, because hydrogen bond make the pairing happens correctly. That is the role of hydrogen bond. Makes the pairing of the bases happens in the correct manner. So when new strains build on template of old strain, so which means this thing, this is the old template, this is the new strain. New strain is this one. Okay. The incoming nucleotides is selected for its ability to pair with base in the old strain. They don't simply pair up, they pair up accordingly. A must pair with T. So how it happens? It happens naturally because they can form two hydrogen bonds at the correct orientation and correct position, and then they pair up. So hydrogen bond plays an important role in the recognitions as different number of hydrogen bonds involved in each pair. So hydrogen bond is important here because they will pair up accordingly. Okay, so we settle already. We settle DNA already. Okay, so how they replicate, have to know, and which one is monomer, which one is polymer, you have to know. Okay, so we settled this, and now let's move on. 12.9 Non-Solvent-Based Adhesives. So which means gum. Okay, adhesive. Like you want to stick two things together, you have to use this kind of adhesive, chemical adhesives. So we have the silyl modified polymer, which is SMP. SMP. So you see how the gum, how the adhesive works over here. So a polymer that contains silicons bonded to oxygen. So you can see the pictures for more details. Okay, you can see silicon. Silicon is somewhere here, bonded to oxygen on top, bonded to oxygen at the bottom. Okay, so it is silicon bonded to oxygen. They're set by reacting with moistures in air. Moistures in air, what they mean by the set. The set means the gum dry up and stick to whatever you want to stick. That is what we call the set. You want the gum to set, you need moisture. You need moistures in the air. That means you need H2O. That's why you see in the picture they put H2O over there. H2O. H2O. So the other words to explain set is the adhesive dries. Okay, if they're dry, we call it set also. We call it set, adhesive dry. Okay, so moistures in the air, which is here. So what happened is hydrolysis happen. So the original structures is OCH3 and OCH3. Because of the H2O, the H2O will go through hydrolysis. That means the water will break down. So when it breaks down, then they will form SiO. SiO will make the adhesive set. And the extra carbons will form methanol. Okay, will form methanol. So of course CH3 come from here, and the water, right? you know, the water, basically what happens is the water will break down to H and OH. H and OH, and then try to imagine, should not be too hard, right? So the OH will stick to one of the CH3 to form CH3OH. Okay, so if they stick to CH3, of course one oxygen should remain here, so that's why there's one oxygen remain here. Okay, so another CH3O together with the O, CH3 together with the O will stick with another H to form the same product, methanol. Okay, CH3OH. So let me put it in this way. CH3O will go and find the H to form methanol. And another one is CH3 only will go and find the OH. And they form the same product, which is methanol also. So because of that, you can easily see that there is one oxygen remain in the structures, and this oxygen will link with that silicones. So they form the siloxanes cross linkage. Okay, they form this. Then the adhesive on the gum dries, and then they will stick to whatever they're supposed to stick. So now, next point. Effectively bonds polymer chains to each other with strong covalent bonds of siloxane cross linkage. So settled already. Okay, so we already finished the first type of adhesive. Now we have the second one, which is called epoxy resin. Epoxy resin. Epoxy resins is examples of thermoset. Thermo means heat, so you need heat. You need heat to make it happen. You need heat to make it set. Set means to make it have the fixed structures. Okay, you need heat to make it have a fixed structures. So let's see, what is that? Polymers that form extensive network of covalent cross-link. Okay, so that means they are polymer that form cross-link. Cross-link is like something like this one. You see the S&P. This is the polymer. The cross-link is here. This is the cross-link. Okay, so cross-link will link between one polymer and another polymer. So that is cross-link. So the cross-link is covalent cross-link. That means it's covalent bond that involved there. Okay, so likewise, this is covalent bond also. Silicon, oxygen, silicon is covalent bond also. So very strong and cannot be melted and remolded. So epoxy resin, once they form a particular shape, so it is very strong, cannot be melted. You cannot think like, okay, we melt it and then we form it to different shapes. Cannot already. Okay? Once they form that shape, it will be that shape all the time. So you cannot melt it down and change it to another shape. So it can't. And formed by reacting monomer with epoxy ethane. Epoxy ethane is the triangular molecule of CH2, CH2O, and a diamides are mixed. Okay, so let's see whether we have any explanation at the back. Yes, actually we have. Okay, so you have to look at the next page, then from there we can understand better. So let's move on. Okay, so let's continue. Just now the notes, they give us some information. They say we have epoxy ethane and we have diamide. Diamide, of course, obviously it is this one. Okay, it is this one. This is the diamide. So you have NH2 and NH2 at the end. That is diamide. So I write down first. Okay, so diamine. Okay, then we have the epoxy ethane. Epoxy ethanes have triangles. Okay. So just now the notes also said it is a triangular molecule. Triangular molecule, which is, you can see here, triangle on at the end. Okay, so this is epoxy ethane. Epoxy ethane. So link with another diamine again. Okay, so now they join up. So when they join up, okay, let's see the details. So diamine is here. Then we have a lot of structures in between, and then NH, and then one of the hydrogen disappear already. One of the hydrogen disappear, and then they link with the triangle. You see the triangle is not triangle anymore. Okay, so the bond is broken already. It's broken. So which means this oxygen is involved. Okay, this oxygen. So 1, 2. Okay, you can see here oxygen, then go down like this. This part remain. And then after that, this oxygen become OH. And then see, okay, they open up. They open up the structures, and then just imagine something like this. This particular part go down, no more forming triangle, but they form a straight chain with the diamide just now. And this one go down and then form a new covalent bond with this one. Okay, so yeah, they form in that way. Likewise, this side also same. This okay, maybe I should put it in this way first. This O, right? This O will change to OH. This O will become OH, as you can see here. Okay, so it become OH, and then this one open down, you pull it down and link with others. So likewise, the other side, same also. This O change to OH. O change to OH. So this O change to OH. And then these structures go down and then form a new covalent bond with the NH2 over here. So that's why you see something like they go straight and form NH and link with NH. Okay, so they join up in that way, and then okay, now let's see. The polymer produced. This is the polymer, this is the polymer. Okay, so the polymer produced forms a giant network of cross-linking with others polymer. Okay, so let's see where's the cross-linking. Okay, this is the cross-linking. Okay, the N with the R with the N, so that is the cross-linking. Cross-linking happen here. So of course, you want it to form cross-linking, right? We need to use heat. That's why it's called thermoset. Thermoset. So we need heat. Okay. So okay. So next one, super glue. Uses addition reactions to stick objects together. So when I say addition reaction, that means initially before they stick together, they have double bonds. Okay, so as you can see in the equation below, you can see double bond is there. Okay, this is double bond. This double bond will open up to form new bond with the next monomer. Likewise, this side will have another bond with the monomer. Okay, so that's why left and right have new covalent bond with others. Okay, so let's see the information here. The monomer is CH2 double bond C, which is this part, CH2 double bond C, CN, CN is this side, and then the bottom here is COOCH3, COOCH3. So it is known as methylcyanol, methyl, and this one, cyanol is CN, acrylate. And addition takes place across CC double bond. So addition takes place here, the double bond. Okay, so polymerization initiated by presence of moisture. Okay, it's the same thing again. No? So as long as you put the glue out exposed to the air, then the glue can set on its own. Okay, now let's move on. This part. Conducting polymers. Conducting polymers. That means they can conduct electricity. Okay, so let's see the information here. Polymers generally insulators. That means they cannot conduct electric. For example, rubber, natural polymers cannot conduct electric. Okay, so but ethane is the hydrocarbon with triple bond. Triple bond. This is ethane, can conduct electricity. Okay, so imagine this is ethane. Now they form polymer, which is called trans polyethylene. Trans you should know lah. The hydrogen is in opposite direction in that way. Okay, so that is trans polyethylene. Polyethylenes. Okay, I think should be easier to take it. The one of the triple bond here, so the electron pair, they put sideways so that you can form new bond with others. So put sideways. So they use one pair of electrons to form new bond on both sides. So that's why they still have double bonds remain. So contains alternate single and double carbon-carbon bonds. So you can see here. So after the triple bond change to double bond, right? So we have double bond followed by single bond followed by double bond. So that is where the double and single bond alternate. Can conduct electricity because p orbitals on neighboring carbon atoms overlap, resulting in long bands of delocalized pi electrons free to move along the chain. So that means the pi electrons is somewhere in the double bond. So they can easily come here, and making the whole chain able to conduct electricity. So that is what we call delocalized pi electrons. So they are originally here, but after that delocalized. So originally here, then delocalize and keep moving. Then doping. Doping is adding other substance, adding other substance. So other substances such as iodine added to improve electrical conductivity of polymer. So this one, if you feel like the conductivity is not good enough, then you can do doping. That means you add iodine inside. That's the process of doping. It's just adding something else into the original one. So that is called doping. And of course, we add with reason. We want to improve the electrical conductivity. Advantages over metal conductors. So why we want to use polymer conductors, we don't want to use metal conductors. So the advantage is do not corrode, which is true. Polymer is not metal corrode. Polymer will not corrode or do not corrode. Much less dense, that means light, light. Shaped more easily. Okay, so example, make into thin sheets to make flat panels that light. Okay, so they can make flat panels. Okay, now let's move on. Okay, so this part is easy. Degradable polymers, that means breakdown of polymers. So first point, disadvantages of plastic. So why they say plastic in the starting? Plastic is basically polymers. Okay, so the polymers, which is plastic, they are non-biodegradable. That means they don't break down by bacteria, by microorganisms. So on combustions, give out harmful vapors. You know, we human beings tend to burn rubbish to destroy the rubbish, but at the end, harmful vapor come out. Poisonous gas come out. So we have biodegradable plastics. Okay, so that means we improve the plastic by changing it to biodegradable plastic. So nowadays our plastic bag is biodegradable by adding starch. Okay, so you can see here, can contain small amounts of starch. You just put starch inside, then bacteria and fungi in moist soil can break it down into smaller compounds, increase surface area, and easier to decompose. That's the reason. Break it down to small part and then decompose more easily. Can be made from monomers derived from plants. Example PLA. PLA is polylactic acid. So right at the side here, this PLA is polylactic acid. This is from plants. We take it as monomer, then we join up to form polymer, then we make it as a biodegradable plastic. And the advantage of doing this is the soil microorganism can digest it easily. Can be hydrolyzed in acidic conditions. That's the advantage of biodegradable plastic. So you put some acid, then it hydrolyzes. Polyamides broken down. Polyamides. Okay. So amides, I hope you know. Still remember CONH. CONH is polyamide. So a polymer with C double bond O and NH, and then link here and there, that is polyamides. Okay. You want to break down this kind of polymer, so you just use acid, then it break down to carboxylic acid and amides. Okay, so imagine the breakdown in the middle. The left hand side will change to carboxylic acid, the right hand side will change to amides. Okay, next one, polyester. Polyester, the structures is C double bond O and link with others. This is polyester. So polyamide, and this is polyester. Polyester broken down to carboxylic acid and alcohol. Okay, so that means you cut in the middle, then the left hand side will change to carboxylic acid. The right hand side will change to alcohols. Okay, so next one, photodegradable plastics. Photodegradable. Okay, what is that? That means, photo means light, sunlight, for example. So you can use sunlight to break down the plastic. Okay, sunlight to break down. But the thing is, we throw rubbish, right? The rubbish is buried under the soil, and no sunlight. So that is the disadvantage. Okay, so carbonyl groups, C double bond O, can absorb energy from ultraviolet wave. They can use the energy. Causes bond in regions of carbonyl group to weaken and breaks. So the bond breaks because of the absorbing of the UV energy. Disadvantages is plastic usually buried under the soil, so cannot get sunlight. If recycled, products are weaker. That means the photo degradable plastic, if you recycle it, is weaker already. So that is the disadvantage, and no more already.